Thanks so much. Yeah, let me start by saying thanks so much, Peter, for a kind introduction. I was blushing. It was very sweet. I mean, really, everybody here is just a fellow colleague, and I'm so grateful for all the collegial interactions and whatnot. It's really important. It's really special. And I'm very grateful for this field because I got into, so I wasn't even planning on talking about this stuff, but I got into this field, I think it's all like 25 or 6 years ago, like middle of the 90s and stuff, when we started doing GC. And never looked back because it was so fun and everything. And there was a lot of opportunities to analyze samples that you could never even dream of analyzing. Like much of Peter's work now, which is pretty outstanding, it's all outstanding, is you're looking at these fuel samples that I'll just tell a story. Like I was at Exxon in 1990, and these people were talking to me, and they said, when you run a 1D GC, what's that hump in the sample? Because they didn't know. They didn't know until John Phillips ran the samples with GC that hump was all these different classes of compounds that if you use 1D GC, you've got all these compounds that come out at the same time, and so the data really is meaningless until you do 2D, right? Especially then with mass spec. So anyway, so thanks so much to the organizers. This is a great venue, and you guys did an awesome job organizing it and stuff, and it's really great. So the other thing I want to say, and I don't know, I just decided I'm going to say it, but so I get to the Seattle airport, and you know, and you just, you know how travel is and stuff. And I just knew that this was going to be a special trip because when I was talking to the agent, they said, your departure date is going to be C18. and it was. So anyway, so C18, we're not doing LC, but I knew enough about C18 from many years ago to know that that's what we were doing. So anyway, so here I am. And we're going to talk, I can probably stand here more. So expanding the scope of tile-based GC data analysis. And so we're really super delighted that LICO has, you know, they licensed this software from us. I don't know how many years that is because years seem to fly by now. But we developed this about 10 years ago, and then they licensed it and stuff. But what this talk is about is the fact that we still have the source code, right? We still have the source code, so that allows us to do in-house development of all the nifty ideas that then places like LICO get to then incorporate it into the licensed software. And so I'm going to touch on some of those developments today. Wait. Oh, that's weird. What's weird is that the down arrow is to go forward. So I got it now. Okay, so anyway, so here's the outline for my talk. We're going to do goals and challenges. We're going to revisit a cycling yeast metabolite data set using basically the chromatography tile. And then we're going to look at kind of a progression of developments here, looking at improvements, I'm going to say, of Fisher ratio analysis with this knee inflammation and Pacu fish. And then we're going to get into some newer things that have been, sounds like they're going to be incorporated into the chromatography tile software that Joe talked about last night with this 1v1 where we compare two samples from each class. And then the whole idea that you can even use tiling to find the main features for data that where you don't know the classes, okay, a priori. So anyway, so let's get going with this. So for the goals of the analysis, and this is all there was this talk about non-targeted analysis. We had a really nice review that came out about a year ago in analytical chemistry. Tim Trinkline and Caitlin Cain led the charge in putting this together. And so you're welcome to look at that. But the whole idea then is that whether you're doing supervised or unsupervised experimental designs, you're trying to take really complex data sets, find, the important data so that you can make actual decisions from it. And all you folks I'm preaching to the choir because all you folks know that this is so important now. Okay, so sometimes you want to identify and quantify the peaks, and so this is where it gets a little bit dicier, like whose software is really doing the best job of deconvoluting or decomposing the data, and then oftentimes these things are translated into targeted analysis methods for routine testing, and there's all these different application areas, and this is just a few that come to mind. And so here's the analytical challenge. You've got 2 chromatograms here and class A, class B, and these samples look the same to me. Okay, and so it wasn't that long ago that people would look at GC chromatograms or LC by LC, and they all try to do it visually, right? And that's just not that. Well, maybe it was a long time ago now, but anyway. But the point is how do you find statistically relevant up or down changes in concentration between 2 or more classes, and do this in the context of overcoming run-to-run retention time shifting, and so we started out doing this with Fisher ratios, and now we're getting into other informative metrics. And so our group also had a fairly long interaction with trying to develop retention time alignment software. Because you might say, well, can't you just align the data? Okay, well, you can align the second dimension data because there's plenty of spectra or points across the widths of the peaks, 10, 20, whatever. But because of the fact that the data is really not sampled very much, due to the sampling density using the modulator, you can't align the data in the first dimension any more than what you have to begin with. And so basically we came up with this this new approach called tiling of the data. It didn't start off that way, but let's talk about standard F ratio. This is the between class variance divided by the sum of the within class variance. So if you have 2 classes, for any given feature, you've got the variance of a given peak in one class relative to the variance of the peak in the other class. And so we see this is just a one-dimensional example. If you're not familiar with the Fisher ratio, class A, class B, even though they're tiny little peaks, they give large Fisher ratios because there's a statistically significant difference in the red ones versus the blue ones. But even though it's a big peak, the first peak and the last peak, it gives a small Fisher ratio. And our first paper on this was, it's like 22 years ago with Kevin Johnson, works at NRL, Naval Research Lab. But that was a pixel-based analysis, and so it was fraught with ambiguities associated with the retention time, misalignment, whatnot. But then it was Brendan Parsons and Luke Marney about 9 years ago now, or 8 probably when they 10 probably when they did the work, that developed the tiling code. And the whole idea is that you want to generate a hit list where the Fisher ratio, the highest Fisher ratios are at the top and the lowest ones are at the bottom. But you want it to be accurate. And just because a code is supposed to do something, that doesn't mean it does do it. So you want to have it, actually confidently provide you with a hit list where the ones that ultimately are up or down regulated in one class relative to the other have a higher Fisher ratio, and the other ones don't. Okay, so the Fisher ratio software, not only is it, in a sense, ranking the hits, but it's telling you everything that's there. So every single solitary peak is found. It just has a lower Fisher ratio. Okay, an insignificant Fisher ratio, in fact. So anyway, so here's some of the papers, more recent to that. Okay, and so I'm not going to spend a lot of time talking about tiling, but basically just the whole idea that this is to address the run-to-run retention time shifting. You pick an appropriate tile size that is big enough to encompass the shifting into 2 dimensions, first dimension, second dimension. You have 4 tile grids, and this might seem like, when we first dreamt this up, this was Luke Marney and Brendan Parsons about 10 years ago. It's like, wow, is this really a good idea? Because you've got all these every single solitary compound could form a maximum of like 9 different hits. Okay, and so that's just an example of one right here. And so you've got, this one happens to have 4 hits based upon where it's falling relative to the tile grid, and then you just have to the software magically finds the location with the biggest star, which is the most appropriate location for it. Okay, so anyway, so this software, it worked. It basically gave you a hit list that really, properly ordered the hits. And so then we had a visit from Lika one day, and Sarah Prebehala was sitting there, and she remembers that day when they stopped by her desk and Sarah typed in, we took a data set, and they were just taken aback by the fact that you could plug in your data into this software, and in 1 minute later, it came up with a hit list that was right for samples that had hundreds of peaks in them. And so then Sarah worked with myself and Liko to take it across the finish line, and they have this delightful chromatograph tile that we learned about a little bit last night, and you've probably already used it. But this is the version that's currently uses the standard F ratio, where you're basically taking the class-to-class variance in the numerator divided by the sum of the within-class variance. And so what you're really kind of hoping on there is that all the variances across all the different samples are kind of they're the same of each other, and you don't have any highly variant peak in one sample. But let's first see how well it worked. And so this is a project that we did many years ago. We collaborated with some heavy hitters in sort of like the biochemical, bioanalytical world, I guess, or biochemistry. And it was looking at yeast, where once you kind of got it going, it would go through this oxygen consumption rhythmic pattern. So that's changing oxygen with time, where basically this yeast strain would grow and kind of partially die and grow and partially die. And then the whole idea was, okay, this is setting off a pattern of metabolites changing in concentration too. And they're not all going to be at the same time because they are linked by these various biological pathways. Okay, and so here's the whole idea is that, we take 24 samples across 10 hours, 25 minutes per sample. You take a sample each dot, and you've got some oxygen consumption profile that might be like that. And then so we could hypothesize that there'd be a bunch of these metabolite patterns that they're not all going to look the same, and they're not going to be all at the same phase relative to the other. Otherwise, it wouldn't be a very interesting project. Okay, and so way back when, you can see the dates, about 15 years ago or so, Rachel Moeller was leading this charge with Ted Young and then Steve McKnight. We collected this data set back then, but our software was, first off, it was a pixel-based Fisher ratio software. It couldn't even do the job. It was horrible. And so Rachel came up with a nifty way of doing it at the time, using a method called max signal ratio. I'm not going to get into how it worked, but it found, and I'll use, say, only 85 metabolites that had some sort of defensible cycling pattern or whatever. Now let's fast forward to just a few years ago, to Lina, sitting in the middle here, not to make you blush or anything, but did this beautiful job when we thought, hey, we've got 24 samples, each one of these points. And so basically we took these yeast samples, 24 samples. So we got this beautiful data set. Let's subject it to chromatograph tile to see if we can validate the chromatograph tile performance in the context of handling so many sample classes. Okay, and what did Lina find out? Instead of discovering 85, I think we discovered everything. 210, way more, 2.5 times as many metabolites. And so it was like, wow, we were just missing all this data. So if you could imagine that you collected some data over the years, it's kind of worth thinking about going back with chromatograph tile now and plugging it in there and seeing what more you find. Anyway, so I can't explain this page nearly as well as Lina can. She's here, so you can talk to her about it later. But here's what those separations kind of look like, and we have some examples here. Myo-inositol. Look at that beautiful. So those bars are basically signals. So a nice cycling pattern, and then there's methyl citrate, high concentration. And so every 12 hours, it was being produced and then boom, consumed by something along that metabolic pathway. So it was a spiky pattern. Then here's this glucose 6-phosphate. We can do some math and find out that it actually has 2 cycles kind of superimposed. And so this is the computational method that's in this paper. And I'm not going to try to explain it, but Lina would do a great job doing that. And there were 672 hits. So what that means is that the Fisher ratio software, nominally the tiling, made a hit list with 672 metabolites in them. Most of them weren't cycling. They were just stuff that, they were real things. They were real metabolites. They just weren't cycling. Okay, and then 210 of them were. 155 of them were unknowns. That's the one. These are all derivatized metabolites with trimethylsilation oxidation. And so the libraries for these things are not complete, especially for the systems this complicated. But this is the heat map from the 55 identified cycling metabolites. So red is the high concentration. So that'd be like the peaks in the myo-inositol, and the blue is the low. And so you can see that we ordered them to make the heat map look appealing, right? They're just ordered by the fact that, okay, the ones at the top, they go up and down, and the ones down here, they go down and up and down and up. And so you got these, and so all of these metabolites have phase differences. And so and then here's the spiky ones, and then here's the ones that had more than one cycle, but it was something defensively real. And so back in that earlier paper, the biochemists had also done some other types of measurements, and then they were able to make sense out of these patterns that we got relative to the biological pathways, and made for some good science. Okay, so the moral of the story here is that the chromatograph tile works amazingly well, with lots of sample classes. It finds everything. Okay, but our job, remember, we have the source code, is to explore other aspects of the tiling capabilities, because it basically solves the problem of the retention time shifting and finds everything. Okay, so beyond the standard tile-based effort, what other metrics can be used? So keep in mind the number of components in your samples is greater than or much greater than the number of peaks in the tick. So the F ratio metric simply ranks the components. The tiling and the hit list generation essentially finds all the components, and the analyze generally utilizes the components toward the top of the hit list in the context of the experimental design. So what other metrics can be implemented to rank the hit list? And so, and this is one of these things where, since we developed this code, you just start thinking like, well, the tiling and the F ratio, they're like connected, right? That was our mindset, that they were connected. And then we were sitting around the lab one day, and we realized an epiphany, right? Like the little light bulb goes on and then it cracks or something, and you realize that they aren't connected. They're 2 separate things. You can tile, you can get all this information, the quantitative data, and then you can put that quantitative data into whatever metric you want. Okay, so that was sort of the epiphany, that they're independent. And so, before that even, we decided, well, there are going to be some applications where you might have one of the, let's say it's a 2-class comparison, where one of the 2 classes, it's like a real juicy analyte that's doing something really interesting, but the variance in one of the 2 classes is kind of big. And that big variance in one of the 2 classes is basically killing the F ratio. It's because it's in the denominator, the within class variance of one of the two is just killing, the value of the F ratio, so you aren't going to find it near the top of the hit list. Okay, and so what we decided to do is the first project, and this is Sarah Prima-Halo's work, basically rank the hits not based upon the standard F ratio and just using the controls for controls versus patients. So sometimes the patient variance would be too high. And for a pure statistician, this is absolute heresy because the F ratio is only supposed to be applied if you have variances that are equal. Okay, and so, but we said, well, we're using this as a ranking metric. And so instead of dividing by the sum of the within class, we're just going to divide by the variance of the control class, which is going to be nice and tidy and small. Okay, so I'll see how I'm doing for time. Am I doing okay? Because I think I think it'll be okay. So anyway, so there's Sarah, and it's a collaboration with the military. And, basically I'm not going to tell you how, this is just in training, like some of the cadets blow out their knee, and so they wanted to know, okay, are there any biomarkers associated with joint inflammation? Can we follow these biomarkers over the course of them trying to heal up and see if they're going to develop arthritis, all this stuff. So we had 30 injured, 30 controlled, and so we do the same thing where these are blood samples of some kind, serum or something, and then, you get the metabolites out of there, you derivatize them. And there's what the separation looks like. There's a ton of peaks, zoom in. And the color coding here is kind of hard, but let's just look at this picture, for example. Okay, arachidonic acid is a really good example. You can see how the signals go from like just above 0 to 1600, whereas the control group, the dark ones here, nice and tight at a low value. And so what you'd say here is, well, some of these patients that I'm circling with the laser pointing, they don't have any more arachidonic acid than the control, so maybe they're not injured so bad. Okay, but then you have some of these bars way out here at a high value. Those are some people that have a lot of arachidonic acid, which is inflammation biomarker. They probably are injured pretty bad. Okay, and so, but you wouldn't want to have missed arachidonic acid as a biomarker just because its standard F ratio is only 13, which is miserably low, and its hit value was, see, I'm jiggling a little bit, but its standard F ratio, that's what that S means, is 123rd, so it's way down the hit list. But then it was hit number 4 using the control normalized, so we didn't miss it. Okay, and I think that example just tells you everything. Interestingly enough, I don't have the data for it here, naproxen was the #1 hit. It was not the #1 hit for the standard, but it was the #1 hit for the control normalized. You know what naproxen is? Aleve. So some of them were popping Aleve and some of them weren't. So anyway, so it's kind of a neat little data. Okay, and then we have other ones that act as almost in situ controls. You have urea that's basically the same in the patients and the control samples, almost the same value, nice and tight distributions. Gives you the nice warm fuzzy feeling that your data is working right. Okay, so now we had another collaborative project with Lina from Argentina, and Lina came up with this idea and the samples to look at fish, these pacu fish that look like little piranha grown in Argentina. And they were worried that maybe the way they were going to raise them was going to impact the quality of the fish. And so, but you could raise the fish 2 different ways. And so the whole idea was that the variance could be inflated in either category. And so we decided, hey, if the control normalized worked here, let's just divide by the minimum within class variance of either class for the purpose of finding the best hits. The proof is still in the end, and I forgot to mention this, but in the end of the day, you still have to do a t-test, right? And so the Fisher ratio software just ranks the hits. Then you have to do the t-test at the end to tell you if it really is statistically significant or not. Okay, so here's the picture here. So they raise these pacu fish, they're Sonia, and then they they used to raise these fish in these aquaria kind of situations, throw in fish food and stuff. And you can imagine that's kind of expensive. Or down in Argentina, where the inflation's like 100% or something. And so they thought, well, let's use these rice paddy fields, And so they said, double duty. We're going to throw these pacu fish in there that have these really nasty teeth. You should look one up on the internet sometime. And so it's like, let's not do this anymore up here. Let's throw the fish in here, and so we can rotate the land. We can raise rice. The fish are in there. We're going to pull the fish out. We're going to sell them at the market. Everybody's going to be richer and happier. But could herbicides and pesticides used in rice farming affect the fish metabolome of these fish? Let's find out. Okay, and so we've got, Lina brought the fish, 10 of each. We went through the sampling protocol, so it was really quite representative of the problem. Okay, so it's not like we had any goofy data here. And back those 2 chromatograms where you can't hardly see anything, we're zooming in on some regions. The point is that we found we just found everything. Again, the software finds everything that means something. So let's just go straight to the punchline here. So we've got a very important column. This is your hit number column. Of course, that's important. F ratios. Remember, this is the MVO, minimum variance optimized Fisher ratio. We're using the smaller of the 2, whether it's farm variance and now this important column is the concentration ratio of a given species farmed over tank raised. And so if anything number less than 1 is probably bad. That means it's something that's lower in the farm raised that shouldn't be lower. Okay, so let's look at 2-butanol. It's associated with a sweet, flowery odor in fish paste. That sounds good. You'd want that to be high. Well, it's about 4 times less than the farmed raise, so they probably don't taste as good. Okay. Now we're gonna look at this gamma one, but let's just move straight to L-ornithine due to the time. It's important for tissue repair and immune response. It's about 33 times lower in the farm fish. So they're getting kind of queasy down there in Argentina when they read the results. Okay. Isoleucine plays a role in purine synthesis, a precursor to aspartic acid. It's important. It's about, 40 something or 50 times lower. Okay. And then last but not least, here's one here. This is a nasty one, 2,3-butanediol. It's 3 times higher in the farm. It's associated with a fishy odor. So it's all this bad stuff. I think you get the idea. I can move on, right? Okay, so and I did give a lecture to the folks down in Argentina, like at their kind of like the equivalent of the ACS for Argentina and stuff, and kind of quiet after the discussion. But 110 analyte hits, 70% concentration ratio, statistically significant. 54 of the 70 of these are important for normal biological functioning and were significantly downregulated in the farmed fish. Okay, and so this downregulation suggests the integrated farming system possibly impacts the pacu fish quality. You can't say it does, you just have to let them decide or not, right? I think we all know that it does. Okay. What's the next one? Okay, so now we're getting towards the end, and I'll try to wrap it up in a minute or two. But so basically, we're all familiar now, I think, with tile-based analyses of Fisher ratio, but replicates may not always be available due to time or expense or limitations. And Joe alluded to this, and Joe, just so you know, I talked to the LICO folks about our work on this, and they decided they were going to incorporate it in their software on their own. And I said, that's all right. That's why I'm telling you. And so we call it tile-based 1v1 analysis. Cain pioneered this one, and it was published about 2 years ago. And now I think you called it fold change last night, right? And so this feature called fold change, it shows up in this paper in analytical chemistry called 1v1 analysis. And it uses the tiling, and we just had to come up with a nifty way to apply the tiling and compare two chromatograms. And also, based upon some work that Grant Ochoa did, not only does it find all the things that you would find with Fisher ratio analysis just with two chromatograms, but it gives you the most purest mass spectrum possible, even better than all the chemometric methods that are out there. And just to show how well it worked, we picked on some data that Liz Humston from LECO worked on when she was in my group about 14 years ago. Okay, and basically we look at cacao samples, and we just took chromatograms at the beginning that were unmolded cacao beans to make chocolate, and molded ones, and then we compared them with this 1v1 software. Caitlin did, not we. Okay, and then we just have boxes around the ones that were upregulated in the unmolded and upregulated in the molded, and so what we're showing here is you might say, hey, I thought you said you only needed one of each. So we're looking at the results of doing this exercise 5 times. So like the first bar, the blue one goes to the first of the red one, second one, just to show that you get consistent results, right? Even though you're just doing it on pairs. Okay, so we've got all these species that have sweet, creamy, citrus-like aromas, that you want to have in your chocolate, are higher in the unmolded, and then they kind of disappear in the molded. That's bad. And then all these nasty ones are higher in the molded ones. And so you might say, well, why does this matter? Well, there's a fine line between fermenting the cacao beans right, that they want to do sort of in situ before they throw them in a ship and take them to wherever to make chocolate, and molding, and so, but they don't know a priori. So sometimes they bring a whole, shipload of cacao beans back, and they're a little moldy. What are we going to do with these? Are we going to throw them out? What do you think? How many think they're going to just throw them out? No, they're going to send them somewhere, and because it's got a lot of chemicals still in there that kind of work for them. They're going to process them. Okay, so they're going to treat them with like sodium hydroxide and throw in some butter and sugar and stuff, and sell them as some of the cheaper types of candy that people like to enjoy. So I always buy the best chocolate, and I have less of it. Okay, so now we've talked about Fisher ratio analysis, where you got a bunch, and they don't have to be all the same. You can have lots of classes, or you can have just reps, that kind of thing. This is showing like blue class, red class, or you could just have reps of whatever. And then we have 1v1, which is the full change. And then we started realizing, hey, all you really want sometimes is you just want to find the peaks where there's a lot going on, right? If you don't know the classes, if there's just a lot of change in the concentration, you want to find those peaks. Okay, and so so same thing, tiling, get the areas of all the peaks, all the different samples, and then we're going to calculate the relative standard deviation. So basically this peak, because there's a lot of range of signals, is going to give you a large relative standard deviation. And so that might be good. That might be informative for the type of chemometric software you're going to use. Okay, so now, Joe, correct me if I'm wrong, but I think you said this is the coefficient of variance method. Yeah, so I talked with the folks at LECO, and I mentioned these papers to them so that, just to keep the record straight on this now. So anyway, so this might be relevant feature in your data set. Okay, so, and so we've applied some of this, and so you could take these and you could pop them in a PCA, not knowing any classes, and find out, look, there's 2 classes, okay? Or it might be a whole bunch of samples that are each different that really would give you an output kind of like this. And they might be finding all the magical peaks to do some sort of partial least squares analysis. And we've done this kind of stuff now, and here they are. There's Paige and Grant and Caitlin again. And here's the project where we spiked in some sulfur compounds and into some, I don't remember what it was, some kind of fuel, and at really low concentration. And then we treated the data like we didn't know we spiked it in. And so we just looked for high relative standard deviations. And there they are, and it's sticking out like a needle in the haystack. There's our spiked in compounds that we didn't inform the data analysis that they were in there. And so then you could do the PCA, and sure enough, there they are. And so that was mostly Paige. And then Caitlin here, she took this data set that we've kind of beaten to death with a stick using before in energy and fuels. She said, well, let's revisit this and let's see if we can get better models with PLS using this method. That was recently published, maybe not even a year ago. Beautiful models. So what are we measuring here? The measured heat of combustion versus the predicted heat of combustion. And you want the slope to be 1, the nice model and everything. And you get these things called linear regression vectors. So blue are the locations of those features that had a high variance that were making, so all the blue ones are associated with making a big net heat of combustion, and all the red ones a low one. Okay, so it's, so when you guys get that coefficient of variance feature, you can have at it and then take a peek at some of these papers. Okay, so thanks so much for, again, for the opportunity to visit and to share this. And so, we continue to kind of work in these areas. And again, we're just delighted to be a part of the community because, the main thing here is to enjoy what we do, train students and do something good for society and that sort of thing. So thanks so much.